Hi, everyone. I think we're ready to get started.
All right. Thanks everyone for joining. My name's Kurt. I'm at Mount Sinai. I'd like to thank the organizers, Dr. Duggar, Flanagan, and Murray for starting this great group of focusing on digital neuropathology. We have a really exciting talk for you today by a PhD student, Armand Collin. He did his bachelor's degree in electrical engineering at Polytech Montreal, and he's just been having this interest in digital pathology and AI ever since then. He studied deep learning in Malaya in 2022 under the supervision of Julian Cohen-Adad. And he's been contributing to this project, AxonDeepSeg, for about 4 years now. And he's really excited to talk about it and show you everything he's done and hopes that maybe you'll integrate it into your own research workflows. So with that, I will hand it over to Armand. And one quick PSA, if anyone wants to speak at this meeting, you can see my email in the side chat, kurt.farrell@mssm.edu. We would love to have you. So take it away.
Thank you, Kurt, for this introduction. So yeah, my name is Armand Collin. I'm a PhD student in biomedical engineering here at Polytechnique Montréal. Let me share my slides. So as Kurt was saying, I am one of the maintainers of the AxonDeepSeg software, which is a free and open source project aimed at researchers. And today I want to bring you on a journey through recent applications of this project. So yeah, I also want to mention Julian Cohen-Adad, my supervisor, who unfortunately couldn't make it today, but he's been involved in this project since the beginning. So my goal with this presentation is to mostly it's to present some recent applications, but of course I'll be giving some context, a little bit about the history of the software, and the main part of part of this presentation will be recent applications. Then we'll shortly go over some future work and what the future direction of this project. So very briefly, here you can see our graphical user interface. AxonDeepSeg is a free and open source software designed specifically for researchers in neuropathology. And what it does is it computes automatically the axon morphometry in microscopy images. Now we support multiple kinds of microscopy, such as electron microscopy, bright field microscopy, or coherent NT-Stokes-Raman spectroscopy. We'll see a little bit more about this last type of microscopy in my slides. Just to give you an idea of what motivated us to create this tool, I'm going to put you in this situation right here. So imagine that you have these two images taken from two different subjects, and you want to compute the axon morphology in both these images. What you would probably do, and what most researchers usually do, is they will take manual measurements on the image, typically in a software like ImageJ, and then they can automatically extract the length of all the radii that they traced in the image. And so this is a very time-consuming process, and you can imagine that just for these two images, you're probably going to have to spend at least an hour to complete all the measurements. And this is only for one radii per fiber, because you could imagine that, for example, in this, you want to compute the G ratio, metric of interest in the field. Very briefly, the G ratio is the ratio of the axon fiber over the full fiber with the myelin sheet. And this gives you an idea of the myelination of a fiber. And this would require then this would require you to take 2 measurements per fiber, so you're doubling the time that it takes. And then another problem is that these are not the images that you collected. The actual images are much larger, and this is what they look like. And you can easily imagine that now, if you want to measure everything in this image, it can take you a few hours, you know. And then obviously, usually you have more than one image per subject, and hopefully more than 2 subjects. So this kind of task quickly becomes impossible to do by hand, and really limits the scale of these kinds of analyses. So this is where our software comes into play. So here you can see we segment the image to know where the axons and the myelin is located, and then we can automatically extract measurements, different measurements for every fiber in the image. And so this process is done automatically. It's very fast to do. Yeah. So I will very briefly present sort of the backbone of this pipeline, but I won't go too far into the technical details, because I was told that it could be it could induce sleep for this audience. But just to keep it very short, the way that we train these models is we need an initial segmentation data set. So here we have the images, and for every image we have a manually generated segmentation mask. And then using these image label pairs, we can train a deep learning architecture to predict these regions automatically. And so we can then produce all kinds of metrics on these images. So this on the right is one of the first applications of AxonDeepSeg. It shows a full rat spinal cord, and so they image this at a very high resolution. We don't see it in this figure, but and then they downsample the metrics, and you can see, for example, the G ratio distribution in the spinal cord, or myelin volume fraction. Here you can see the axon density. So these are the kinds of analyses that we're interested in here. And now I will go over a very brief history of the software, just to give you an idea of how this project started. And so the first date that I put here is 2015. 2015 marks the publication of the UNED paper. So this might ring a bell for some of you, it might not, but this was a pivotal work in biomedical image segmentation, and it is still to this date, 10 years later, state of the art for biomedical image segmentation. And now this is the underlying deep learning technology behind AxonDeepSeg. In 2016, our group released AxonSeg, which was the first iteration of this software, and it was also the first open source tool for axon and myelin segmentation. So you can see that the history of this task goes back, almost 10 years, and our lab has been involved in this since the beginning. Now in 2018, the AxonDeepSeg was released, so we can see it as a spiritual little brother of AxonSeg, and it now used this UNED technology. In 2019, I put this here. This was sort of one of the first one of the early applications of the software, and so what they did was they imaged human spinal cord, so over 23 actual slices covering C1 to L5 spinal levels, and they generated the first comprehensive mapping of axon morphometry in the human spinal cord using AxonDeepSeg. So I thought this was an interesting application of the software. Very briefly, in 2022, one of the previous maintainers of AxonDeepSeg integrated this new backend into the software, and she also contributed to a standard for our microscopy data sets. As you know, open data is a very big issue in biomedical image processing. It's a bottleneck for a lot of researchers to access the data. So we've also been focusing a lot on how to share this data, the best practices, and how to standardize these data sets. And finally, in 2024, we integrated the new backend again. So maybe some of you have heard of NNUNet. It's a framework that's developed in Germany by a group in Germany called the Division of Medical Image Computing of the German Cancer Research Center. And to this day, it's a state-of-the-art for training your UNet models. This is not what we use. And this is what the UNet looks like. It's a convolutional neural network that takes as input an image and outputs, in our case, a segmentation where the axon and the myelin are identified. So a typical collaboration for us, what it looks like, as I was saying earlier, the initial overhead of these projects is to train a segmentation model. So we typically have to collect some samples, some small amount of data, essentially, and iteratively train a model with bigger and bigger amounts of images. So this is what we call the active learning technique, where we first train a small model to segment on more images, then we correct its predictions and retrain it on a larger set of data, and we can iteratively grow the data set while keeping the manual human intervention at a minimum. So before going into the recent applications, I wanted to quickly acknowledge some people. Stoyan Asenov, who's been working on our graphical user interface for some years now. Mathieu Boudreau, who's sort of a long-term maintainer of the software. Julien Cohen-Haddad, of course, my supervisor, and Marie-Hélène Bourget. So Marie-Hélène Bourget worked on this before I arrived, and yeah, she was a very crucial part of this project. So let's get onto the recent applications. I feel like this is what's going to be the most interesting. So I'm starting with this study that was done. It started in 2021, I think, with Professor Osvaldo Del Bono from Wake Forest University, and in collaboration with the Rush University. So the data we used here came from the Rush Memory and Aging Project, which is a longitudinal study that focuses on common chronic conditions of aging, including decline in motor function. So this is what they wanted to quantify. Our study included 140 participants, non-diabetic participants, and for which we quantified the axon morphometry in the postmortem tibial nerve. So here you can see on the left one of these beautiful image. It's a nerve cut from a 84-year-old participant, and the middle panel shows a magnified view of just a small region of interest, which we can compare on the right to the associated segmentation mask, with the myelin in gray and the axon in white. And so measurements are automatically extracted from these masks. And yeah, so I just want to say that the publication detailing our results is currently under final review. It should hopefully be accepted this year. But yeah, this study was made possible by AxonDeepSeg because we analyzed a lot of images Now, the second project I want to talk about, this one I didn't work on this, but I think they definitely showed some very interesting metrics there. They did some interesting comparisons between the measurements obtained from AxonDeepSeg compared to manual measurements. So this study was done by Simeon Dashler from Sick Kids Hospital in Toronto and Marie-Hélène Bourget from our team. So it resulted in a publication in 2022. And so for this study, they were interested in nerve regeneration. So they had nerve samples from 27 adult female rats, including 4 tibial nerves, 6 median nerves, 17 common peroneal nerves. And yeah, as I was saying, the data comes from an experimental nerve repair study. So they have early regeneration and late regeneration time points where the axon density and the axon morphology changes. So we included all these we included images from all these different scenarios in the training of the model. And this was imaged using light microscopy at 63x magnification. So here we can see some of the examples from their analysis. So here you can see, for example, a healthy nerve. The segmentation looks quite regular. And then for the early regeneration nerves and late regeneration, we have a different picture. So early regeneration nerves have a lot less axons, and they are usually they are somewhat deformed. And this is also a little bit the case for the late regeneration nerves. And so on this image on the right, you can see, I want to bring your attention to these 3 graphs here. So these are the axon diameter distributions for the healthy nerves, the healthy regeneration and late regeneration nerves. And what they did was they compared in blue the measurements from AxonDeepSeg to, in gray, manual measurements. And you can see that although there is a slight tendency of AxonDeepSeg to underestimate the axon diameter, which is pretty consistent across the 3 groups, the general trend is very much represented in the AxonDeepSeg measurements. So yeah, I thought this was a pretty good example of what AxonDeepSeg can do. And yeah, the last thing I want to mention about this project is the analysis time. So here we can get an idea of how much time this can save. So what they did was they measured how much time it took to manually measure the fibers in an image. So this was only for a 100 and 100 micrometer ROI. And so the scale bar right here is 100 micrometers. So it's a very small part of this image. And it took, on average, about 20 minutes for the annotator to complete his measurements. So obviously, depending on, here you can see on orange, blue, and green, the different groups. So obviously for the early regeneration, there are less axons, so it takes less time to measure it. But on average, it took about 20 minutes, whereas for AxonDeepSeg, it was essentially instantaneous. So I think it most certainly would take less time for AxonDeepSeg to measure everything in this image than it took for the annotator to just take measurements in a 100 by 100 micrometer squared ROI. So yeah, hopefully this gives you some perspective on as to how much time can be saved and the sort of the scale of the analysis that this enables us to have. Now, the third project I want to mention is this collaboration with Professor Alan Peterson from McGill, and for these results were published in 2024. But this study was they investigated this protein called the myelin basic protein and its effect on the sort of myelin dimensions, myelin features. So for this, what they did, this is a typical example of the kind of studies that we assist. They used different mouse lines that accumulated different amounts of MBP mRNA levels, ranging from 8 to 160% of the wild type levels. And they collected images from the spinal cord, 3 different regions, and also from the corpus callosum in the brain, using electron microscopy. And so here you can see just the relative MBP mRNA levels in the optic nerve for all these different mouse lines, and we can see that they all accumulated different amount of it. And their question was how does this affect myelination? So this is what they did using AxonDeepSeg. We can see that just looking at it visually. Here you have for example, the spinal cord time points, and every row represents a different mouse lines, and we can see that the morphology obviously changes across all these groups. But then they had so many mouse lines, so many regions that they were analyzing. It would have been very hard to do this by hand. So this is why they used AxonDeepSeg, and they were able to basically quantify these morphology differences between all these mouse lines. Now onto an ongoing project that the results are not yet published, but this is an interesting study in collaboration with Valérie Pinot-Noël from Laval University. Her supervisor is Daniel Cote from a Cerveau Lab, and also a collaboration with Kelly Perlman from McGill. So she is supervised by Professor Naguib Mekawar at McGill. So Naguib Mekawar is also the director of the Douglas Bell Canada Brain Bank, which is it's the largest brain bank in Canada. So they coordinate brain donation and prepare the samples and send them to researchers. So this study focuses on the lasting impact of child abuse on oligodendrocyte lineage cells and myelin. There's a focus on a specific region of the brain, which is the uncinate fasciculus. So the UF is it's only found in human and non-human primates, and it's also the last fiber tract to completely mature in the human brain. So to study this phenomenon, they wanted to compare the myelin morphology between different groups. So they have a depressive with suicide group, and they also have a depressive suicide with child abuse group, as just to basically isolate the effect of child abuse from the effect of depression. So it's a very interesting data that they have here. But the problem is that it's a pretty challenging task to image this part of the brain with CARS microscopy. Now we didn't really have a choice to use CARS microscopy because the samples are sometimes in a pretty rough shape, and staining them, or the tissue preparation required for optic or EM microscopy would be too damaging. So they had to use CARS, which doesn't require any staining. So it uses the natural properties of the myelin, for example, to show contrast in the image. So they used the same subject as this 2017 study from the same group, and yeah, the exact same subjects. So in this 2017 study, they found that they found a significant difference in G ratio, axon diameter, and myelin thickness for the group with child abuse. So this is sort of the motivation why they wanted to then move on to the onset fasciculus and study this phenomenon in a different part of the nervous system. And you can see here some examples from these images and the segmentation. So it's a pretty challenging task, but we're slowly getting there and gathering a lot of fibers per subject. Yeah. Now, the last projects I want to talk about are with our Stanford collaborators. So this first one, so all these projects are in collaboration with Professor Bradley Zucchero from Stanford University. And so they identified a transcription factor that is essential for myelination. And as such, they had an interest in including unmyelinated axons in their analysis. So this is something that we didn't support prior to this collaboration, but we essentially added the option to quantify also the unmyelinated fibers in the image, which allows us to produce this metric, which is the percentage of myelinated axons. So this is because this transcription factor has a very profound impact on myelination. They are very interested in this specific percentage. So we were able to produce this percentage automatically using AxonDeepSeg. So in this study, again, it's a study where they have different mouse lines. They generated an SRF conditional knockout mice, and where the SRF was conditionally deleted from OPCs using olig-2-Cre. And now they mostly want to compare the percentage of myelinated axon between control and this knockout mice. And for this, they use high resolution TEM images and this technique called high pressure freezing and freeze substitution, which creates these beautiful images that we will see in a minute. But other structures are segmented as well. So we also decided to segment the oligodendrocyte nucleus and processes because it helped us just to get a better precision for the axon segmentation. So yeah, I just wanted to show this very quickly. This is one of the image that they acquired, and the resolution is actually so it's actually also pretty absurd, but we can see the individual myelin sheets surrounding the axons. So we don't need this amount of details for the software to work, but this is obviously welcome, and it's a very cool thing to see. So these figures are taken from their 2024 paper, and we tried to reproduce their results, but using only AxonDeepSeg. So here we can see this is the graph that I want to direct your attention to. It's the percentage of myelinated axons, and you can see that they found a significant difference between the two groups. Actually, we can kind of see it just visually here. The knockout mice they have a lot less myelinated axons. We can see the unmyelinated fibers everywhere, whereas the knockout mice have a just regular looking axons, and we were actually able to reproduce their results. So here I'm showing just very quickly what our data set looked like. Here you can see a magnified view of these images where I highlighted the myelinated fibers in green and the unmyelinated fibers in pink. And on the right here, we can see predictions from the model for myelinated and unmyelinated fibers. And so if I just if I show you this magnified view right here, you can see that the unmyelinated fibers are located a little bit everywhere in the image. And using AxonDeepSeg, we can automatically segment these fibers. And this is the number of axons that we were able to score using AxonDeepSeg here on the right. And on the left is the number of axons that were scored by the annotator for the 2024 study. So a couple of things I want to say here. The person who did this acquired these manual measurements was we could say a little bit zealous, and they took on average around 5,000 fibers per mouse. So this study had 10 mouse, and this is much higher than the standard for these kinds of studies. But yeah, so this person took a whole week of doing only this task full time, and using AxonDeepSeg, we can do this in under an hour. So yeah, you can imagine that this opens up new possibilities for researchers. They can waste less time doing these kinds of tedious and repetitive tasks and focus on some more important stuff. And so yeah, very quickly here, I'm showing this percentage of myelinated axons in the lower row here. These are the measurements, the manual measurements, comparison between the wild type and the knockout mice. And here is the comparison that we had with AxonDeepSeg. So essentially the same level of significance, and yeah. So this is something that we want to use on other projects now, because we want to make this feature more robust, and I think we think that it's a metric of interest for a lot of other researchers. And talking about other applications of this method, we have this other project with in collaboration with Duke. Yeah, so for this project, they were interested in a different protein called the inverted formin 2 protein, and its role specifically on the actin disassembly. And for these for this purpose, they wanted to compute the axon morphometry and compare it between the wild type and a INF2 knockout mice. So yeah, so for this they use SEM images of the optic nerve. These images are scanning electron microscopy, but just so you know, the contrast was inverted, so this is why they look like this. And we were able to segment. I'm only showing the myelinated fibers here, but we also segment the unmyelinated fibers, and we can measure a lot of different metrics. So this is still an ongoing project. I'm not gonna go too much into the specifics, but yeah, the final project I want to talk about is this collaboration with Madeleine Cooper and Maya Weigel, again from the Bradley Zucchero's lab at Stanford. So they had an interest in the FGF family of proteins and its role in morphologic changes to oligodendrocytes in vivo. So for this purpose, they generated FGF receptors 1 and 2. They generated double conditional knockout mice where the FGF receptors 1 and 2 are inactivated. And using this double knockout mice, they are able to study the effect on axon density and disruption of myelin ultrastructure. So again, they use the TEM imaging and high pressure freezing freeze substitution. So it gives you these images, and I have this is a magnified view of these images. So this is still an ongoing project. We're interested in how this how FGF basically impacts myelination, and AxonDeepSeg will allow us to do this analysis automatically. So I will very quickly go over future work, and this will be the last part of the presentation. So this is an idea that we've been working on for a few years now. So one of the person I worked with on this was Arthur Bochet from which he was an intern in our lab. So the previous AxonDeepSeg versions had one model per modality. So we have one model for transmission electron microscopy, one model for bright field microscopy, and so on. And the problem is that it becomes hard to maintain a lot of different models on the long term. And we wanted to kind of unify all these segmentation model into a single monolithic one, which would be easier, hopefully easier to maintain, and also easier to use for AxonDeepSeg users. And so here you can see a bunch of different data sets that we collected over the years from different modalities. On the top row, you have EM. On the bottom row, you have bright field and coherent anti-Stokes Raman, spectroscopy. And the modality is not the only thing that changes across these data sets. We also we have a lot of different species: mouse, macaque, rat, human, dog, even cat, rabbit, and sometimes different pathologies, obviously different resolutions. So we have a lot of variability to show the model. And yeah, so I will not bore you with the details again, but we were able to segment all these different modalities, all these different domains using our a single model, which we call the generalist model. And it's now integrated into the latest version of AxonDeepSeg, thanks to Arthur and also Mathieu Boudreau from our team. And the last project I want to talk about very briefly is this project that we've been working on with Muriel Mardenly and Edgar Kapov. So they were mostly interested in axon detection. So instead of segmenting the axon fibers, we would only detect them. So it's an object detection task, and this would be useful for studies where you only need the axon count or the percentage of myelinated axons, like we were talking about earlier. So these kinds of studies where you're mostly interested just on the count and maybe axon density, these types of metrics, you're not necessarily interested in the morphology. These models, the object detection models, are much easier to develop than the segmentation models. So we thought that it would be very interesting to have to offer this feature to our users, because these annotations for these models are much less costly to obtain. So you can imagine we only need points or bounding boxes, as opposed to having a manually corrected segmentation mask, which is not only time consuming to do, but I have done a lot of it, and I can tell you it's a horrible task. But yeah, so this is still an ongoing project. And I put this slide here. Maybe the next collaboration could be you. So we're always happy to take a look at your histology images. Always happy to discuss what's possible and how our software can be used, how we can help you. So yeah, with this, I think I'm done with this presentation. Thank you for your attention. And yeah, I put some links here. If you want to get in touch with us, we'll be happy to hear about you. Thank you.
Amazing. That was such a good presentation. We have actually quite a few questions and comments
Right.
that we'll go ahead and get started and ask. I'm going to start the ones that were placed into the Q&A section somewhere in the chat as well. So the first question is from Addison Berg. It is, what is the data size that goes into training the tool? What is the variability in the axonal myelin integrity or health in the training data? And how does the tool deal with demyelinating pathologies like MS or CIPD?
Okay. So the amount of data that goes into the tool is it's pretty variable. We've had success in training models with only like 2 or 3 images, and it worked very well because their data set was kind of homogeneous all across. But in some cases, of course, it gets a little bit more complicated. In these cases, we always help the researchers to build the data set, and we help along in the process. But yeah, for example, you're talking about demyelinating diseases, which cause morphology changes. So in this case, we usually need examples from all the scenarios. So if you only show the model healthy axons and then you present it with demyelinated axons, it's not going to know what to do with these new cases. So this is something that we always keep in mind. We have to have images in the training set from all these different scenarios. But yeah, so I hope this answers your question.
Great. It's a great answer. Well, the next question is, can you elaborate a little more on the fixed sampling and native sampling method in your training regime?
Right. So we used to resample our training images to native. Okay, so just to clarify the question a little bit, the native resolution. So the images are acquired by researchers typically at a wide range of magnifications of actual pixel sizes, because depending on the microscope, the specific microscope you use, for example, a 63x magnification will not always lead to the same pixel size, right? So we have we essentially have different pixel sizes for every data set that we collect. And what we used to do was resample all these images to a single resolution. So you can imagine if you have images at 0.2 micrometers per pixel, we resample them at 0.1 micrometers. So you're going to change the dimensions, the physical dimensions of the image. But we're moving a little bit away from this right now because we're trying, as I was saying, to unify all our segmentation models. And in this process, we're trying to have a model that works at all these different resolutions. So this is still a work in progress, but I can say that we're trying to sort of mitigate this problem. So yeah.
Great. Actually, in the chat, the person asks the question wants to ask a follow-up question. I think that's
Sure.
fine. Go ahead and unmute yourself and ask.
I thank you for the great presentation. I really liked it. I was just Wanted to just go over the part of the fixed and native sampling again, because this is something that even I have encountered in our research, that the model that is trained on a fixed sampling will only perform good at that sampling resolution. So like I saw in your architecture, I think you just pass both the images together to the model. Is that what you're doing to counter that change in sampling? Or I don't know, maybe you could elaborate more on that. That would be great.
Yeah, so the way that we're trying to mitigate this is, first we only show, we do not do any resampling before the training. We just show the original we keep the original image dimensions. But the direction that we're going towards right now is having very aggressive data augmentations that will sort of like change the resolution pretty aggressively of the image at training time, so that the model can see a very, very wide range of resolutions. And we've had some preliminary results that indicated that this works pretty well. But if you want to discuss this further, I'm happy to discuss this over email. You can send me an email, and I'll be happy to share with you an exchange on this specific topic.
Sure, thank you.
Thank you.
Very question.
Next question is, have you been able to compare your algorithm with Fiji, binarizing, and watershed particle counts in terms of accuracy?
Yeah, so we didn't really compare it directly. Well, the first iteration of our software did something very similar to what you were describing. So it was based on non-deep learning segmentation methods that used a mix of thresholding and a lot of different interesting engineering tricks to produce the segmentation. But the problem with these is that they don't really I mean, the development of these methods is like very, very project specific. So in some projects this could work very well. I have seen some images that look very, very clean and very regular, where you could apply some thresholding and do this and obtain very good results. But the problem is that sometimes I think some of the images I showed during the presentation, this would be impossible because there's too much, the SNR is not high enough, essentially. And also, I would say that these kinds of techniques are sometimes very effective, but they're not very automated, because you usually have to find the good threshold for your image. Sometimes the contrast varies between different images, so you might have to use a different threshold for every image. So yeah, this deep learning allows us to not care about all these issues and basically show a variability to the model, and it reacts to it. But
Awesome. Then the next question is, any obvious speed ups or switching to object detection? What was the trade-off in terms of accuracy and speed compared to the segmentation model?
Yeah, so this is still an ongoing project. We don't have like we do have preliminary results that show that the accuracy is very similar to the segmentation models, and it could easily surpass it. But the thing is for object detection, you only you're limited to certain metrics. For example, you can compute axon densities, you can compute axon counts per subject, or the percentage of myelinated fibers, but you can compute the axon diameter, you can compute the G ratio for the fibers. So yeah, this is a limitation, but it would definitely be a speed up. Let me just finish by saying this. one of the limitation of our segmentation models is that we're able to run them very, very efficiently on GPUs or very good computers, let's just say. But on our users' computers, sometimes it can take very long. In some cases where they have old laptops, we've seen it take seen the segmentation process take a long time, and this is where object detection could help, because these models are much lighter, much more friendly for your CPU, and so on. Yeah.
Amazing. The next question is, thank you for sharing AxonDeepSeg. What were the biggest challenges for developing AxonDeepSeg and publishing as an open source tool?
Okay, so let me preface this by saying I was not like the original developer of AxonDeepSeg. I wasn't involved at the very beginning, almost 10 years ago, in this project, but I can tell you that yeah, developing such an open source tool is it has some definite upsides, and sometimes it's difficult to share the work with others, and I would say that I have heard that in the beginning, when they published the first AxonDeepSeg paper, it was a bit hard for them to find a journal to publish, because at the time open source was not as much, let's say, it was not as much promoted. But I will say that the times are changing, and the sort of climate right now is very favorable to the open source community. Yeah.
Things have changed. Great. Next question is, I understand that you have the same architecture for each modality. Is the architecture of your generalist multimodal segmentation also exactly the same? If yes, how do you deal with the domain shifts in staining, label space, etc., and change in resolutions? Was there a dedicated module for that?
So no, there are no dedicated modules. Our approach for this project of a unified segmentation model was to just basically more on the data side, that on the model side, we tried to get as much variability as possible, as much different species, as much and closely follow the segmentation on all these different scenarios. So our long-term goal would be to have a model that can be continuously trained on images, on new images. So if you have a new pathology or a new domain to show the model, we can just create a small data set and fine-tune the model on this. This is the ultimate goal that where we're sort of converging towards. But yeah, this is still a work in progress.
Thank you for that answer. I had asked that question earlier too. So I just wanted to confirm again, this is still a unit architecture with a lot of data augmentation for the generalist model, right? Not a foundational model or anything like that.
Yeah, exactly.
It's surprising to me that a unit model is also, performing this well. Great job to the team.
Yeah,
Thank
yeah. Well, the unit architecture is very surprising technology. As I was pointing out, it was developed 10 years ago, and on the deep learning side, nothing lasts 10 years. But on the specific subject of biomedical image segmentation, we have some limitations, which I won't really get into, but we do have some challenges that the unit really mitigates. So yeah, thank you.
Thank you. I'll email you more about that.
All right. And next question is, can you elaborate on the architecture of your unified model? How do we find out more about your implementation? Details.
Right. So we submitted the preprint. I can find you the link. so we have a preprint, but since then we started to we changed the implementation a little bit, but hopefully this would give you an idea of everything we did here. I think this is the one. Yep. So I'll just post it in the chat.
Awesome, thank you. And this is a quick follow-up question. How does your method overcome the contrast limits?
Yeah, so the contrast limit, I would say, is not the biggest bottleneck, because you can just show, you know, images to a model that have two different contrasts. If you have enough images, it's gonna know what to do with all these. And there also would be other strategies, like for instance, you can invert the contrast in some cases to match the training data. But yeah, can you repeat the question?
So
How does
I was just, yeah, this was me, Ina Vano, UCLA. I just was wondering, because you had mentioned when you compared it with the Fiji algorithms that they're more rigid. I mean, there is a thresholding that is local, taking the local environment, for instance, into consideration. But I'm sure your algorithm must have had some flexibilities to account for changes within a section, for instance, in the brightness, dimness, contrast in general. And I wanted to just hear your opinion how it's overcome.
Yeah, so for example, for the brightness, these issues are usually mitigated by pre-processing. So we usually put, you know, equalize the histogram of the image and do some basic pre-processing that sort of standardize what the image look like. But of course, it doesn't account for everything. And yes, I will say that the over the years, my philosophy on this is if you ever encounter a situation where the model has is not able to predict correctly in your images certain regions for these you know, in terms of contrast or maybe staining, you can very easily correct this in its behavior, meaning that we can target the specific area where the model is most uncertain and do some corrections there and retrain the model or just fine tune it quickly. So I would say that, yeah, this type of technique is it can be it's pretty flexible when it comes to this, because then once you show it once to the model, it knows for every other images that look like this, right?
Very cool. Thanks so much.
All right,
Yeah.
I'm going to give some rapid fire ones here, so keep the answer short. This first one can just be a yes or no. Will your software segment cells as well?
I mean, yes, we do for some cells, but just to keep it short, no. If you're too far from axons and myelin, there are other groups that actually work with these, and feel free to get in touch with us, and I can redirect you to more specialized groups.
Great presentation. Is there a way to differentiate between myelin and fibrous tissue or other pathologies?
Yeah, so this is an area of investigation for us. Are there some ways to are there some ways to differentiate the, you know, pathologies at a large scale and identify them? So I would say it's still a work in progress on this side, but yeah.
Well, fantastic. We've reached the top of the hour. I think a lot of the other questions were somewhat answered by some of the other answers that Armand had given. So if there are any other questions, feel free to email him. And if you're interested in being a speaker, feel free to email me at kurt.farrell@mssm.edu. So thank you everyone for coming. Thank you, Hannah at the NACC, and thank you, Drs. Duggar, Flanagan, and Murray for starting this wonderful seminar series. Have a great afternoon.
Bye. Thank you, everyone.